Bismillah Rahman Rahim. Hello and welcome back, friends. Welcome to another video lecture by Kami Biology. In a series of video lecture, we are going to talk about bacterial cell wall or prokaryotic cell wall in a detail. And so this is actually the video lecture regarding with the cell. And in this video lecture, what we are going to talk about, in this video lecture, we are going to talk about bacterial cell wall. Bacterial or prokaryotic. Prokaryotic cell wall. Now this topic contain four or I think 3 or 4 videos. This topic contain. In this first video, we are going to talk about the overview of bacterial cell wall. Means overview. Now this is the overview part of the bacterial cell wall or prokaryotic cell wall. First of all, what is a bacterial cell wall? A bacterial cell wall, it is actually a layer which is come after the cell membrane. Let's suppose if this is the cell membrane, layer. So after that, there will be another layer which comes. So this another layer are called what? This another layer are called the cell wall, and this cell wall is actually responsible to give shape. This cell wall are also responsible to protect the cell from osmotic pressure. This cell wall are also responsible to prevent the cell from toxic substance like drugs and some other toxic substance. And it is also important to improve the pathogenicity of the cell wall. Why? Because this cell wall is just like the capsule. And inshallah, I will also make a separate video lecture on a capsule. Means the cell wall are also responsible for the pathogenicity of the bacterial cell. Now, before going to explain the cell wall, first of all, we need to understand that how the cell wall is actually looking in gram-positive and in gram-negative bacteria. We have actually 2 types of the bacteria, gram-positive and gram-negative bacteria. Or we can also say gram-positive cell wall and gram-negative cell wall. Actually, we have 2 cell wall. These techniques or the cell wall which are present inside the gram-positive and gram-negative bacteria, actually it is, the bacteria is actually divided on, means gram-positive bacteria and gram-negative. We have 2 types of the bacteria, gram-positive and gram-negative, on the basis of the cell wall. In gram-positive bacteria, we have a thick cell wall. And in gram-negative bacteria, we have a thin cell wall. Okay, but what actually happened? A scientist named Christian Graham, I don't know the exact name, Christian Graham. The scientist's name was Christian Graham. In 1884, a scientist named Christian Graham discovered a techniques called Graham staining techniques. Due to these techniques, they can identify the gram-positive cell wall and they also identify the gram-negative cell wall. And after that, they can say that, and after that, they can also discover the gram-positive bacteria and gram-negative bacteria. I am not explaining the Graham staining techniques in a detail, but what actually happened in Graham staining, the gram-positive bacteria will be look like purple if you watch it in the microscope, and the gram-negative bacteria will be look like pink if you watch it in the microscope. The gram-positive bacteria have a thick cell wall, and the gram-negative bacteria have a thin cell wall. Okay. Now, if we see in gram-negative and gram-positive bacteria, so how the cell wall will be look like? This is, let's suppose, the outermost layer, and this is what this is actually the peptidoglycan. So in case of gram-positive bacteria, so they have a thick peptidoglycan layer, thick peptidoglycan. So they have thick peptidoglycan layer, and they have also a cell membrane. So this green layer represents what? This green layer represents the cell membrane. So the cell wall, or we can say the bacterial cell wall, or we can say the peptidoglycan layer will become after the cell membrane. So this is what? This is the plasma membrane, or we can also call cell membrane as well. So this is very thick, and the thickness of this cell wall or peptidoglycan will be, so the thickness of this peptidoglycan layer will be 20 to 70 nanometer. So this will be the 20 to 70 nanometer, it will be thick in case of the gram-positive bacteria. So in gram-positive bacteria, the cell wall will be thick, means this peptidoglycan will be thick. But what actually happened, this cell wall are also responsible to protect the bacteria from osmotic pressure. To protect the bacteria from osmotic pressure, due to which the bacteria will be not lysis, because the peptidoglycan layer are very strong and very thick. Now there is also a space you can see, we simply called it periplasm. We called it periplasm. This space is actually called periplasm. Okay. Now this is a little bit about the gram-positive bacteria and the cell wall in the gram-positive bacteria. Now we have also a cell wall or peptidoglycan layer in gram-negative bacteria as well, but it is quite different. How? Because in gram-positive bacteria, first of all, we have the plasma membrane, and after that we have another, and after that we have a peptidoglycan layer. But keep it in your mind that this peptidoglycan layer in case of the gram-negative bacteria will be thin, but in gram-positive bacteria, as you can watch it, as you can see it, it is thick, but in this case it will be thin. And after that what we have another. Now you can easily see this is the cell membrane. What we call it, this is the plasma membrane. Now this will be the inner membrane, and this will be the outer membrane. Due to this way, the peptidoglycan layer will be thin. So this is what? This is the peptidoglycan. Peptidoglycan. Now the thickness of this peptidoglycan may be 7 to 8 nanometer. They will be thick 7 to 8 nanometer. So this will be the thickness of this peptidoglycan layer. It means it is very thin in this case. And as you can see, this is the space between them. As you can see, this is the space. Means the space, as you can see. We simply call that space, we simply call that what? Periplasmic space. Periplasmic space. In gram-positive bacteria, the space is called periplasm. Why? Because it is filled by this thickness of this peptidoglycan layer, and in gram-negative bacteria, this space is called periplasmic space. Why? Because it is not filled by any kind of the protein or by this peptidoglycan. If you see, the peptidoglycan is very thick, and there are some proteins, but there are some proteins. As you can see, these proteins are called exoproteins or exoenzyme as well. These proteins are actually responsible to degrade what? To degrade the large polymacromolecules, and that's why we call that periplasm, not periplasmic space. Why? Because it is already filled. Means the space is already filled by the peptidoglycan layer and some other proteins. But in this case, the space is called periplasmic space, and it is not filled. But here just a very thin layer of the peptidoglycan are present. So that's why we call that periplasmic space, not periplasm. So, okay, so this is very important. This cell wall, or you can see this peptidoglycan, we also call that cell envelope. envelope. And it is just like the capsule. Okay, I will also explain the capsule in some other video lecture, not in this video lecture. It is also just like a capsule, and it also improves the pathogenicity of the cell. Fine. So this is about the bacterial cell wall or prokaryotic cell wall, the overview of the prokaryotic cell wall. Inshallah, in the next video, we are going to talk about the peptidoglycan structure in a detail. Fine. So please stay tuned and continue to watch the rest of the video. Thank you so much.